Thanks again for everyone coming. I would say that this is the largest audience that I've had for my presentation. And so I will combine all my 3 small sessions into a big one. And if you have questions, I'll leave some time for you to ask before the coffee break. So I'll talk about 4 parts in my session: visualization, data integration and alignment, denoising, and transfer learning. So the first part is visualization and dimension reduction. So we're going to talk about what we're measuring for single-cell RNA sequencing. So if we summarize what we get, one representation is this heat map of gene expression levels for each cell. So each column is a cell, and each row is a gene that we measure. And this number can be the number of reads we get for the gene, or the number of copy numbers that we capture for that RNA. And so for each cell, you can review these tens of thousands of gene expressions as its feature space. And typically, we can measure tens of thousands of genes at the same time. So that feature space is really huge for the cell. And when we're doing single-cell RNA sequencing, the first thing we want to know is what is the cell population that we're measuring, and how we can visualize the cells that we get from the technology. But a cell has tens of thousands of features. So this dimension reduction or visualization is that how we can find, if for visualization, we may want to get like 2 or 3 features of each cell, and then we can see the cells in a 2-dimensional, 3-dimensional map. Or for dimension reduction, we may want to reduce these tens of thousands of features of genes into, say, like 20 or 30 dimensions. So we can do downstream analysis like clustering or pseudo-time construction much easier, and remove some noise that we see from the data. So for visualization, we want to turn from this heat map of the cells to this 2-dimensional map. Here in this map, this is a t-SNE plot, and I'll talk more about what is a t-SNE plot in the next few slides. And here, each dot is a cell. So you can see that from the visualization, we can see that these cells have different clusters. And I want to mention one thing here is that these colors for the cells are determined by a separate clustering algorithms that Rhonda mentioned earlier. And if you have more interest, there are a lot of talks online you can see about how to do single-cell clustering. So we won't talk about that in detail in this tutorial. So this is how. So you can. For here, I put this color so that you can see better how the cells are. You can see how the cells are grouped into clusters by visualization. So if you have learned some course on statistics, one thing you may be familiar with for dimension reduction is using PCA. And PCA, basically what it's trying to find is it's a linear dimension reduction tool, which means that, say, this is the higher space. So here we can only see 3 dimensions, but actually your data can have tens of thousands of dimensions. And this PCA is trying to find linear subspaces. Here it's just a 2-dimensional linear space that can represent the variations of your data as much as possible. And here, we are trying to find this 2-dimensional space that can represent the variations of the data as much as possible, and this is what you will see through the PCA. And so so you project all your cells in the high-dimensional, all your samples in the high-dimensional space to a low-dimensional, 2-dimensional linear space in this sample. But for single-cell RNA sequencing, people find out that these cells are not just lying in low-dimensional linear space. They find out that nonlinear dimension reduction tools actually typically works much better for visualizing the cells. And 2 popular methods that I will talk about today is first is the t-SNE plot that you have just seen, and the other is UMAP. And this UMAP, this benchmarking paper, single-cell RNA sequencing, just came out last year. So it's kind of a quite new software that people are trying to apply on single-cell RNA sequencing data. So the idea of t-SNE is that it tries to capture the distance of cells in the original space, and to represent that in a lower dimensional space. And for high dimensional space, so how to calculate the distance between two cells? One popular method, for example, what the PCA is looking at is the Euclidean distance. But people think that for single-cell sequencing data, the distance between two cells that are nearby to each other is more informative than the distance of two cells that are far away to each other. So we want to, in the low-dimensional representation, we want to, the t-SNE can capture more of the local distance between cells. And how they do it is, instead of just using the Euclidean distance in the original space, they calculate the distance between two cells, I and j, by this exponential transformation. So you can see, so this exponential family has a decay function of the distance like this. So if the distance is if the distance between two cells is very close to each other, it will weight more than if these two cells are very far from each other. And so, so this distance measure try to capture the local distance more. And in the low dimensional space, this SNE, which is stochastic neighbor embedding without t, is we also try to we also measure 2 points in the low dimensional space by this Gaussian density function, which is through some exponential transportation of the original Euclid distance. And how to find the low dimensional space is by minimizing this KL distance between these two spaces. But then people find out this stochastic neighbor embedding is not very useful. It does not always work very well, because in the low dimensional space, it's throw away too much the distance that are kind of moderately close, but not too close. So they replace this Gaussian distance function of the distance transformation to a more heavier tail distance function, which is the density function of the t distribution, so that it will give a higher weight to the distance of the cells that are, it will keep moderate far away points, not too close. And so that's how it's called t-SNE. And this UMAP is like a new method that people are trying on single-cell sequencing data. Like the t-SNE only keeps local distance of the original, of the cells in the original space. But then people think that maybe the longer distance also has some meaningful information. So this UMAP can kind of still focus on the local distance between cells, but it will still give some weights for the cells that has longer distance. So the idea is based on a complicated topological theory in mathematics, but I will just talk about the high-level ideas. So the first step is it try to find a manifold of the original high-dimensional space of the cells. Well, on this manifold, the cells kind of uniformly locate on the manifold. And they constructed that manifold by using the k-nearest neighbor graph. For example, if in a high-dimensional space, the cells lies on a ring, it will try to find that ring. That's the manifold that it tries to find for the cells in the high-dimensional space. And then And then the second step is that they try to represent the manifold by low-dimensional points. And that is based on some complicated theory of fuzzy topological representation. I will not talk in detail because it's not really helpful for us to understand if we don't have a solid mathematical background. But compared with t-SNE, remember that in t-SNE, it tried to measure the distance between two cells in the low-dimensional space using the density function of the T distribution. And here it's just replace the density function of the T distribution by a more general form. And these A and B are determined by the algorithm. So here, this is a graphical representation. So this is kind of the high-dimensional original points of the cells that has tens of thousands of features. It tries to find a manifold, and then it tries to represent such a manifold in the low-dimensional space. So why people develop? So these are figures from the original UMAP paper. These are not single-cell sequencing data. These are some imaging data sets. But I will show you how these three methods perform on single-cell RNA-seq data in the next slide. And here, just give you a brief idea of how these three different methods work. So the last row is the result of PCA. You can see that PCA cannot separate different clusters very well. And this is the results of, so these are three, so the columns are three different data sets. And the first row is the result of UMAP. And the second row is the result of t-SNE. And so I think they try to convey the message that because UMAP and t-SNE, they just keep local distance, so you can see clusters of samples or data points or cells in the single-cell sequencing data better. But this UMAP, it tries to kind of also keep more local, global distance features. So you can see, for example, here, like globally, it looks more similar to the PCA plot. For example, here, it starts from so here you can see some orange and red together here, so they're still together here. And you see some like yellow and green together here, so stuck together here. So kind of this UMAP, it keeps local distance, but it looks more similar to the PCA, which also keeps the global distance. And this is a result of PCA, t-SNE, and UMAP on the same single-cell sequencing data. Again, these colors are determined by a separate clustering method. It's just help us to kind of judge how good each visualization method is. And I like apply a standard serial pipeline where you can see the tutorial from their website on the same data set. So you can see here that if you're using just PCA, you cannot see the clusters of cells very good. I think one problem with UMAP is that they do give you some like shape that is really arbitrary. But I think here it's not very apparent, but seems that this is more similar globally to the PCA map. And if you can, so this is the data that the UMAP paper provides. So you can see, so they try to advertise the effect of UMAP, so you can see the effect more clearly here. So because it keeps the UMAP clips global distance, so you can see that these two populations, they're both CD8 T cells, and they're still kind of close to each other in the UMAP, but they're like really separated in the t-SNE plot. That is what they advertise. I think in practice, you don't always see that. So, but one like very beneficial thing for UMAP is that it's much faster than t-SNE. So you can try it when you have very large scale data. So if you still remember the t-SNE, you need to calculate the cell-cell distance. So it goes square like with your your number of cells. So if you have n cells, then the cost, computational cost, is O n squared. But in UMAP, they have some very clever way to construct a manifold so that it grows almost linearly with the number of cells. So this is the data size, and this is kind of the computational time on some measurement. But I just want to show you that, of course, like PCA is the fast because it goes linearly with the number of cells. But UMAP is kind of comparable to PCA, while t-SNE can take a long time where you have, say, millions of cells. So that's why people like to try it. But you do see kind of, I think it's also here, like it has some like shapes that is kind of just artificial due to the UMAP method. So that's the first part about visualization. I think t-SNE is currently the most popular, and people are starting to use UMAP. And I also want to mention that here for visualization, you just keep the data to 2 or 3 dimensions. But if your dimension reduction is the first step for next downstream analysis, then you can keep whatever dimensions you want, like 10 or 20 or 30. How many dimensions to keep is typically a hard question to determine. I think this software, they have some default methods. And in practice, you can just try different number of dimensions to see how it affects your downstream analysis. So the second part is about data integration alignment. So this is related to the batch correction section, batch correction part that Rhonda briefly mentioned. Here we have more time to talk about it. So I hope to answer the questions that some people just asked before about batch correction. It's not a simple method that you may have thought for single-cell sequencing data because of confounding. And I just introduced some methods that people have tried. So what is data integration or alignment? I have a simple cartoon. So say you have like several different data sets. For example, you will measure the same tissue, say the kidney tissue on 3 different samples, three different individuals. And you are thinking that, well, these are all like healthy people, and you will see like common cell types in these three people. However, if you do, say, if I just merge the data and then I just treat them as one data set, when you visualize the data, it's not always happens. I think, for example, 10x, it has less batch effect. But in a lot of data sets, you will see that these cells are separated by batches or individuals. And you don't like this result because, for example, I think that these 3 people, they share many common cell types. So what I hope is that, if I see this, it looks clear that there are 3 cell types. The first 2 individual, they just have 2 of them, and the third person have a unique cell type that is not representing the other 2 individuals. So this is what we want to see. And the goal of data integration is we want to turn from these kind of visualization results to these visualization results. So why we see unalignment between data sets? So first, there are biological differences. When you are measuring different individuals or different tissues, for example, you may have cancer tissue or normal tissue, there are different cell populations because there are different individuals. And also, maybe some individual have a different cell type that is not representing the other cell populations. So you also see unalignment between data sets. And also, in one of these data alignment papers, they also try to align human mouse cells. So you know that they are different. So, of course, you don't see them aligned to each other. And that's the biological differences. And some of these biological differences are what we're interested in. For example, if different individuals have different cell types, then that's kind of what we want to see from the single-cell sequence data. But some of the biological differences are the things that we are not interested in. For example, if you measure 10 different people, their cells may be different just because of like they have different age or different so there are features that we're not interested in. So that's kind of one difficulty because like there's no clear cutoff on like what we're interested in, what we are not, or and so kind of that's one difficulty. And also, the unalignment is also typically due to technical differences. And that's the difference that we are not interested in. For example, batch effects. If these data are processed in different batches, they're just different because you have different bias in different batch. And also, these different data sets, maybe one data set is highly it has a very deep sequencing depth. The other one is very shallow. So for one data set, you will see like 99% of the entries at 0. The other data set, just like say 50% of the entries at 0. This is just due to different sequencing depths. And that's also something that you're not interested in. That's for different data sets. And it's data integration and alignment a batch correction problem. If our goal is to remove the technical differences, then that's kind of a batch correction problem. And and people also use it to remove unwanted or not interesting biological differences. For example, for mouse and human, there are so many differences, but they still try to align to see like maybe if they are maybe just interested in what are different cell types. What is a new cell type in human that is not represented in mouse, and they're not interested in the common difference between human and mouse. So they also try to. So that's a kind of a very vague goal. Though in the papers, the researchers want to do that. And but I want to mention here is that the biggest issue, the biggest challenge is they are confounding between batches and unknown cell types. For example, when I try to align two different samples, they are different because they are in different batch, and they are different also maybe because one population has a new cell type that's not represented in the others. So I want to. So that can be characterized in statistics, the confounding between batches which you know and the cell types in the different populations that is not known. And as someone asked previously, can I just incorporate the batch information into a model? So I think that cannot solve such a confounding issue. So that's kind of people want to try to use these graphical methods to solve it. So these are current methods. They are all very new. Some of these methods are already published. And so I will focus on these 3 methods in the tutorial. And there are a bunch of other methods. Their goal is to kind of just to make the method run faster on very large data sets. And these other 2 methods, they are kind of not just exactly for data alignments, but I still list it here because they can kind of do alignment or remove batch as a side product of the method. So the first method I will introduce, maybe the earliest method for data integration. It is called MNN correct. So I want to mention that these 3 are also mentioned in Rhonda's introduction slides. I just give you more details about the high-level ideas of these methods. So, so the idea of MNN correct is I try to find pairs. So let me expand here. So here, the cells that are, these are for one batch, and these other cells that are like kind of transparent, semi-transparent are for the other batch. And they try to align these two data sets. And these colors, you don't know them when you do alignment. So that's the difficulty. That's the unknown cell type that you don't know beforehand. And so, but you know the batch information as this is the data that so you know the batch information because that's the information that are given to you when you have the data. So the idea is to use the some kind of nearest neighbor methods. So they try, so for one, so the basic idea is simple. So for one cell in batch 2, it wants to find a pair in batch 1. So they think that these two cells are actually kind of the same, but they are different just because they are in different batches. And in the second batch, they try to find a pair in the first batch. And but I said earlier that there can be different cell types in different batches. So their idea is that I just try to align the cells that I'm for sure that kind of their pairs. For example, if the pairs, so they find the pairs by nearest neighbors. So if the pair in the other batch is the nearest neighbor for this cell in batch 1, and this cell in batch 2 is also, so the nearest neighbor in batch 2 is, let me try to explain it. So it's for this cell in batch 1, if its nearest neighbor in batch 2 also has itself as the nearest neighbor, then kind of they are like mutual nearest. So then I I have some confidence that these should be similar cells and they are just different because of batches. But if this cell, it has the nearest neighbor in batch 1, but its neighbor is not, it's very different from itself. So then I think, well, this is maybe some cell that is not represented in the other batch. So kind of, then they try to identify, well, these are the cells that kind of I'm very confident who are whose pairs. Then I try to merge them. And then for the other cells, this may be just the unique cell type that is represented in one batch but not in the other. So that's the idea of MMM correct. Okay, then like this multi-CCA, so it has two versions. The first version does not perform, people later show that it does not perform really well because I'll say why, like kind of briefly, because their second step is a little bit ad hoc. But the second version, they combine their idea of CCA with the MMM correct idea of using the mutual nearest neighbors, and they kind of do like very well in most data sets. And so, but I'll first introduce what is the core idea in multi-CCA, which is finding this CCA, which is the canonical correlation analysis, I guess. I forgot the full name. But it's a little different from the CCA in statistics. But so I'll try to expand their idea with this graphical, with this matrix multiplication equation. So these are two data sets. So they can align multiple data sets, but this is a cartoon illustration of their idea when you have two data sets. This is from batch 1, this is from batch 2. They share the same genes, but these are different cells. So the rows are different cells, the columns are the genes. So their idea is that, well, I think this might maybe just from two different individuals, but they share a lot of common features. Like, for example, they share common cell types. And these are the parts that they share. So they share, so you can understand this as, for example, there are 5 cell types that are common. And these are a dimension of 5. And for each row, it is the gene features for that cell type. So they share some common gene features, but these cells are kind of, they have different like projection on this shared gene features. But of course, these these cells also, these data sets, they also have some unique things. For example, they have different batches, and also they have features that is not shared. They also have the technical noise. So the first step is they try to, from the data set, they try to calculate the gene feature space B. And then what they get, when they calculate B, then they project the original data into a low dimensional space W1 and W2. And the second step, so these rows are cells, so you have a new representation of your original cells, but they are like on the shared gene feature space. Then the second step is they try to align the cells on the using the these two matrices, W1 and W2. And in the idea of CCA1, the second step is kind of very complicated, and they don't have a good reasoning for that. So they change, so I'll omit the detail of that, but in the second version of multi-CCA, they make this alignment, so they make this step to be more like formal and use the idea, borrow the idea from MMM correct. So I think that makes a lot of sense. So the second version of multi-CCA is, I would say, it's a hybrid of multi-CCA version 1 and MMM correct. And so the first step is CCA. Now they're looking at the lower dimensional space, W1 and W2, and you can think like this is W1, this is W2. So they have the cells in the first batch and the cells in the second batch. And they kind of, on this lower dimensional space, they use the idea of MMM correct. So they find anchor cells, which are the cells in MMM correct that are like, I have confidence that it has a pair in the second data set. So they try to find these anchor cells, and they do some anchor scoring by like judging like how many nearest neighbors are there. Let me see how I can explain that. So they score each cell to like kind of determine how likely they are an anchor cell by determining so if these two cells, so for example, if these two cells, they're kind of nearest neighbor to each other, if they're nearest neighbors in the second batch are also kind of nearest neighbors, then they think that, well, these are cells that it's like similar cells in the other batch, and these are kind of anchor points. And if these two cells, for example, if these two cells, they are nearest neighbors in the second data, it's very different, it's very far away, then these may be the cells that are like kind of don't have a good representation in the second data, and these are not anchor points that I want to rely on. So they find these anchor points, then they align it based on the anchor points, and then they can align all the cells. So that's kind of, these are technical details that are different from MMM correct, but these are not the core idea. And they also can do label transfer and feature imputation using multi-CCA, but that's not really related to data alignment. So in this they show their performance. So these are different batches, and these are the cell types. So I want to merge these different batches, but I still want to keep different cell types separate. And you can see that, so this is another method of data alignment that I did not mention. And so you can see that there's Surat version 3, they can merge these colors the best. This Surat version 2 and MMM correct, due to some reason still, you can see separations of different batches. So this, for example, these methods, they rely on an assumption that did not, important assumptions that your batches cannot be too large. So if your batch is kind of too large, dominating the difference of cell types, then they will make mistakes. So these will rely on your assumption that your cell type information is very strong and your batch effects is not too large. So they can still correctly find your pairs in the other data set. And you can see that they show that throughout version 3, they can separate these different cell types, while they still are not doing perfect. But this is the result from multi-CCA version 2, so it may have some biased towards the result of their method. Okay, so the third part is denoising. So first, as Rhonda mentioned earlier, that single-cell RNA sequencing data is very noisy. How noisy is it? So this is an experiment that can kind of give you an idea of how noisy the data is. So this data is released in this paper, Cell System paper in 2018. So they do the fish, so both single-cell RNA sequencing and RNA fish on the same melanoma cell line. So these are the same cell population, but they are measured by two different technologies. And single-cell RNA sequencing, they measure 8,000 cells, and it can measure the whole transcriptome. So it have 12,000. It measures 12,000 genes. And this RNA fish data. So this is kind of like a figure of what is RNA fish. So it looks at, it has imaging-based technique, and these are the RNAs. They can kind of count how many RNAs you see in one cell. And they measure actually much more cells, but this technology, currently cannot give you the whole transcriptome. So this data set, they have 26 genes. I did some filtering, and I just keep like 11 genes overlapping and kind of have high confidence that their measurement is kind of good. So there are 11 genes that I keep that overlap between fish and dropsy. So these are the same cell populations, so they're measuring the same things. They're measuring the number of RNAs in one cell for one gene. So you would say if these two both these two technologies are perfect, then you would see like their measurement to be very similar. So they are not measuring the same cell, but they're measuring the same cell population. So the population based, so for example, the distribution of their measurements in the population, they should be similar across the two technologies if both technologies are perfect. And we kind of, I think they're saying that the RNA fish is much more accurate than dropseq. So kind of we're using it as a gold standard to see how noisy dropseq data is. So how we look at it is we look at distribution-based properties of a gene across cells. Because as I said earlier, they're measuring the same cell population, so you would think that this distribution features, they would match between the two technologies. So I look at three different things. One thing is the variation of gene expressions across. So the sequencing depth is a lot bit it's different. So because of the sequencing depth issue, so I normalize the single-cell sequencing data, and all these three features, they are invariant of scaling, so they are still comparable between the two technologies. So first, I look at the variations across the cells between the two data sets. So for one gene, first I look at the variations of their gene expressions. And so here, there are two measurements I use. One is CV, I think kind of you're familiar with CV. CV is variance divided by, it's a standard deviation divided by mean. And the Gini coefficient is kind of another measure of inequality of the population. So both these two are measuring the variations of gene expressions. And the x-axis is RNA fish, and the y-axis is single-cell RNA sequencing, and each point is a gene. It's one of the 11 genes that are overlap between the two technologies. So you can see that the single-cell RNA sequencing data is much more variable compared with your measurement using RNA fish. And this third measurement is looking at the fraction of zeros. And this non-zero fraction is 1 minus the fraction of zeros. And you all can also see that the single-cell sequencing data has a lot more zeros than the RNA fish data. And this gap is because you have much larger technical noise in single-cell sequencing data because you have because it is more noisy, so your measurement has larger variation and has more zeros, but these are not the real variation than the real zeros that actually exist in your cells. So this is Rhonda talk about the protocols, and this is just the single-cell sequencing workflow that I download from Wikipedia. So you can see that from cell isolation to the final single-cell expression profile that we get, it goes through these many steps, and each step can bring technical noise and can bring measurement error. And when they aggregate together, the technical noise is really huge. So, so the idea of denoising is to remove the technical noise. So here, so people also call it imputation, but I would like to call it denoising. People call it imputation because you see a lot of zeros. Many of the zeros are dropouts. They are not the real zeros, the real no gene expression in your cell. And when you denoise, you have much less zeros, so kind of you impute the zero. But I would like to call it denoising because the non-zero entry is not a perfect measurement. It also contains the technical noise. So you're not just impute the zeros, you're not leaving the non-zeros still there. So you're kind of denoise, you are removing the noise in all entries. So I would more like to call it a denoising method. I think that's a more accurate description of these techniques. So, this is a kind of one snapshot of how denoising can help. So this is visualization. So this is some HPB PBMC data. And if you do not do denoising, when you visualize the cells, so these are the cell type labels, you can see the T cells are mixed together. If you do denoising and then visualize, then you can see the T cells are much better separated. So it kind of makes your cells a cleaner cell. And it also can improve recovering the gene expression patterns. And these are some known marker genes of these 5 different T cell types. And you can see, for example, these are the marker genes of the regulatory T cells, but because the data is very noisy, you do not really see. So these are the heat maps. If it's a marker, then that means that express more in one cell type, but not the others. But you do not see it very clearly because it's too sparse. That the data is very noisy, and after denoising, you can see the patterns much clearly. And these are the current methods. So it's also, these methods are kind of also very new. So I would focus on more on, because kind of people are using deep learning methods, trying deep learning methods on single-cell sequencing data. It has some benefits, and it's kind of a hot topic. So I will focus more on the deep learning approaches using autoencoder. For other methods, you can see, if you're interested, you can see their papers. So I think the first, the paper that I think describes best the kind of the how autoencoder is used in single-cell sequencing is this DCA model. So this is what they show. So the autoencoder is kind of a nonlinear dimension reduction. So if you use PCA, you have your each layer are colored in different colors, and for your input, so these different circles are different features. So this may be like 6 different genes, and your cells are the samples. So the idea, for example, when you're using PCA, is like you project your high dimensional space to a low dimensional space and project it back. But using autoencoder, the idea is that I do a nonlinear dimension reduction to reduce the tens of thousands of genes to a bottleneck layer of, say, like 32 nodes. And then I project it back to the original space. And the idea, and the reason they use autoencoder is that for single-cell sequencing data, people think that, as I said earlier, linear linear dimension reduction is not working very well. And so your input here is the original noisy single-cell RNA sequencing data, and your output, so the loss function is determined by, like, I have a idea of the technical noise. So they think that you observed data is a zero inflated negative binomial, follows a zero inflated negative binomial distribution of your true. So this mean mu is they think it's the true gene expression, and you also have some other extra parameters. So originally you have a noisy data, and you hope that your output here, the mean mu, is the denoise output that is a cleaner data set. So graphically, they're thinking, so I think this is a nice explanation. So these blue dots are your observed data. But they think that the true gene expression lies in this low-dimensional manifold, which is like the projection from your bottleneck layer to here. So they're trying to find this manifold, and these are like white circles are your denoise data. So the difference from this with dimension reduction is that for dimension reduction, you are satisfied with finding, say, the bottleneck layer in some way, but you're using other methods like t-SNE. And here you denoising, so your input has tens of thousands of genes. Your denoise data still have tens of thousands of genes, so you want to map it back. So that's the idea of DCA. And the SCVI, it is using another type of autoencoder. It has a similar graph, but it's using the variation autoencoder. Well, here the bottleneck layer, they have some randomness here. The details I won't talk more, but you can look at their paper. The variation autoencoder is a popular type of autoencoder in the deep learning literature. And so in SaverX, so it's because here you think that your true gene expression lies in a low-dimensional manifold. That's actually not really true. People find out that there are biological variations, true biological variations in your gene expression. That's noisy, but it's has some it's functional function related. For example, there's one paper saying that when the mice gets older, the variation of gene expression of the immune cells gets larger, and that says you have less control of the immune system. So in SaverX, our method, so we still want to keep such kind of biological randomness. So we try to separate biological randomness from the technical noise and still preserve the biological randomness, which we think are related to some functional properties of the cells. And so that's a kind of, it's not assuming it lies in a manifold, but still has some randomness. But for details, I won't talk about the details. So that's all these three methods. You can see that they don't differ too much in terms of using the autoencoder. So one thing of using the autoencoder is that these are actually very fast. So if you really have millions of cells, and so these are deep learning methods, but it's not too deep. So typically it's just 3 or 4 layers. So if you just have a CPU instead of a GPU, you can still train it. And if you really have millions of cells, they can finish in maybe, I think, 20 or 30 minutes. It's not too much. And compared with so some of these other methods, they take longer time, or they don't perform as well. And, so one problem, so in our SaverX, one problem we try to solve is that, so for this deep learning methods, it has millions of parameters. If you kind of know a little bit, have read some literature about deep learning, because from each node to this node, the parameters, there are 2 parameters related to like one line. You can see there are thousands of lines, because here this may be like thousands of nodes, and here these are hundreds of nodes and like 36 nodes. So there are 2 parameters for each line, and so it can aggregate when you can easily have millions of parameters. If you have millions of cells, then it's okay. But sometimes you don't have too much cells, then you cannot really train the deep learning, the autoencoder well. For example, even you have tens of thousands of cells in the kidney, maybe I'm just interested in the immune cells in the kidney. Then that will just be, then when you collect, you will just see that you will collect just hundreds of immune cells in one kidney. Then how can you denoise these hundreds of cells? When you put it in your autoencoder, it does not really work well because you don't have enough samples. And then that starts my fourth part for transfer learning. Is we want to borrow information from external data to help us understand better of our small target data. So why? So transfer learning is a kind of idea in the deep learning literature. So the origin of that is that, so for example, you have Google, they have 10 millions of pictures of cats or say animals or cars. So they can train a neural network super well. But say I'm just as a customer, I just have hundreds of pictures of cars. These are maybe just different cars with from the cars pictures in the Google storage, but they have some similarities because they are still cars. If I just trained a deep learning model using just hundreds of my car pictures, it will perform really bad. Then the idea is I want to transfer the information. Google already learned about the cars in the Google storage to help me understand, say, classify better the car pictures, the hundreds of car pictures of my storage. So that's kind of the idea of transfer learning. And for single-cell sequencing data, why we still think transfer learning is a very promising idea. So first, you may know this Human Cell Atlas project. It's a very influential project. It's a global, so the goal of Human Cell Atlas is that they want to create a reference map. Well, they want to measure for all cell types, all major cell types, and all tissues, and to create a reference map. Well, for each cell type, for example, for each tissue, for the kidney tissue, they measure 10 people, and for each people, there are 10,000 cells. So you kind of have a map for the kidney tissue, but that's for these 10 people. And they want to learn ideas from these healthy people, but in the future, this reference map may help you understand better the disease population or some other people that are not in the map. It has similar idea as the Human Genome Project, that you have the genome of 10,000 people, and that will help you learn the genome of other people. Here, you have the profile of the cells of 10 people. That may help you understand the profile of cells in other people. But it's harder because in the Human Genome Project, your DNA sequence, there are not too much randomness. The RNA expression, there are a lot of randomness in different people. And that's kind of, but people still think that this Human Cell Atlas project it gives you an idea of what the tissues look like to be very beneficial. But how to use that in the future is still an open problem. And also, people also have created mouse cell atlas. These 2 papers, they are influential papers. This paper, they have measured 500,000 cells, mouse cells in 40 tissues. and this Tabular Morris Consortium. Their paper, they have measured 10,000 cells and for 20 tissues. So kind of, they already have created kind of blueprint of the cells in the mice. And the question is, will these cells in the mice give us some information that we can learn about human, since the human cell atlas is still not yet done? You all know there are differences between human and mice, but they all share some similar cell types. So can we still learn important information while remove those information that are just unique to the mice? So the transfer learning idea in SaverX is we want to pre-train. It's very easy. So the idea is just like so remember that we have an autoencoder that has millions of parameters. And if we just have hundreds of cells, it's hard to train them. But I can collect publicly available data that has similar cell types. For example, I have the immune cells in the kidney tissue example. These human cell atlas, I have already collected 500,000 immune cells in the cord blood and also not from the kidney, but they are immune cells. So these immune cells have similarity with the immune cells in your kidney, in the kidney. So I can first pre-train the autoencoder with this kind of public data. So I already have some weights that are already determined by these data sets. That's a pre-trained autoencoder. When you have a new data set, so this, you can also pre-train it with other species like mice. When you have a new data set, you just train the autoencoder again with your new data set on top of the pre-trained autoencoder. Because the autoencoder is a highly over-parameterized model, so while you initiate the model, your optimization actually matters. It's just this transfer learning is that you use your public data to get a good initiation of when you optimize the autoencoder with your own data. And this other translate has a similar idea. And for details, you can look at the paper. And the transfer learning is not just used for denoising. People are trying transfer learning also in other types of problems. And this SC gene, they want to, so they're doing a very interesting thing, but that's kind of also a hard problem. For example, you can, so these are different species. So these are control and these are after you stimulate. You can imagine, so here they're just using, these are rat and these are mice, but you can imagine they have it on the animals, but these are human. So you can do experiments on mice, but you cannot, it's hard for you to do experiments on human. So what they want to know is that, well, I do all this similar simulation on other species, can I predict what's a human? For example, here is rat, but for you can also imagine you can do it on humans. So I have the healthy person. I want to predict what their cells will look like when I perform a stimulation experiment on human, though I cannot. So I don't have this data, but I want to predict what the data will look like with given the control data and also this stimulation data from other species. So this will be super useful if they get very good results, because this just means that you don't need to really worry about doing experiments on human. And so the idea, but what they already done still depend on strong assumptions. So their idea is the high level idea is like this. So let's just focus on this. So these 4 data sets, actually 3 data sets, because this one you don't have the data set. So you have the control mice, you have what it looks, so these are some cells, a group of cells. And you have what it looks like after you do the stimulation. And you also have some healthy, the control cells of rat. and the goal is to predict what this group of healthy the red cells in control group will look like when you add the stimulation. So their idea is that they use the variational autoencoder. So what the variational autoencoder can tell you is they can find a map. So if you have two groups of cells, they can learn a map from one population of cell to another population of cell. So you may have heard some news like in the imaging problems. Like I have a picture of cats, and after some mapping, I can turn the picture to a dog. Or like I have a picture of a female, then I can just do some mapping, so I can map this female to a male person. So this kind of people use in CS they use some, for example, GAN, but variational autoencoder can also do that. So this kind of style transfer. So the idea is that they first transfer this to mice, and then they learn how you transfer. So these are a group of cells. So through the variational autoencoder here, they can transfer it back to a group of cells of mice, and then they can do a map from a group of cells of rats of mice under control to a group of cells of mice under stimulation. Then one big assumption is that they assume the transformation here is the same as the transformation here. Then they transfer the stimulated mice using the same transformation to the stimulated rat. So the big idea is that they assume that different species have different response. So they think that the response here is the same as the response here. So that's a big assumption because they're assuming that if it's about human mice, you're assuming that your human has the same reaction as mice, which is still unknown. But this kind of is a first try of style transfer in single-cell RNA sequencing. I think one big problem of applying these deep learning methods in single-cell sequencing data is that we don't have very clear gold standard. For example, in the imaging classification problem or some other problems, like they already have like a group of pictures that they know very well which are they already know very well the classification of these pictures. Or like kind of they try to denoise a picture. You can just visualize. So this is the original picture and what it looks like after denoising. But for this single-cell sequencing data, your gene expression profile, you cannot easily visualize it. So you don't know really well like how good you perform because you don't have a very good gold standard data. But on the other side, the single-cell sequencing data is using deep learning methods is promising because, for example, if it's bulk RNA sequencing data, if you have like say millions of 10,000 people, that's already a huge sample size. But here, with the single-cell sequencing technology, you can easily get millions of cells or even more, which means that you have a large number of samples. It's like the imaging problem that is successful using deep learning. So it's still promising using deep learning methods in single-cell sequencing data, but people are still trying different methods, not very, very solid. I cannot say very confident that deep learning works really well in single-cell sequencing. There's still a lot of room for that. For example, in this paper, I think their problem is very interesting. Their solution is not perfect, I would say. And this is another work that is done in our group. It's not done by me, but done by my colleague. So they try to transfer the information of the surface protein prediction. So you may know there are techniques. So Yuchao will talk about multimodal data. So this statistic data they measure both single cell, for one cell, they measure both the RNA expression, the whole profile of the transcriptome, and they can also measure the like tens of, maybe 40 like surface proteins. So this surface protein is less noisy, and it will be very helpful in identifying the clustering these different cells. But that technology is not available for all single-cell sequencing data. And the idea is that, so I can learn a mapping from your RNA expression to the surface protein using the Statistical data. Then when I give you another, for example, the Statistical data, they have the immune cells, the PBMC immune cells. Now I have the human cell atlas data, which only have the single-cell sequencing data, but don't have the surface protein. I may use this map that I learned from the Statistical data to predict the surface protein in the human cell atlas. So that's the high-level idea. And in their paper, they show that it works, and it still keep. So the main thing is that we need to guarantee that we still keep some unique feature in the new data set. But in their paper, they show that with the predicted surface proteins, you can cluster the single-cell sequencing data, the human cell atlas data better. So that's the thing that you learn from the Statistical data. That's the thing you transfer from the Statistical data. So that's all of what I want to talk. So it's the time for questions. There's still 20 minutes left.
Basically, for the batch correction, you explained two methods, but in the table you show that there are other available tools for this. So what are the differences, and main question, how to select the most appropriate for the task?
Oh, so here these are current methods. So I have a little bit of feature explaining. I think, for example, this ligger, there's also based on, so I think basically either they use the nearest neighbors, either they use like CCA, but the details, the technical details are different. And that can, the actual algorithm is different, and that can affect the speed of the software. I think currently people may think the multi-CCA method is very promising, but I think there's no agreement on which method is best, and there's no benchmarking paper yet. So I think I would not know what method to be used in real data, but at least I think multi-CCA version 2 is better than multi-CCA version 1. Yeah.
Hi, very nice talk. Thanks.
Excuse me, can I just add a few things based on the previous question? Okay. So I'm going to talk about the CCA in my section a little more in details, but simply based on that question, my understanding is that for the CCA, and the MNN, I agree with what Jingshu said, that there are two different methods. They try to do the same type of thing, which is try to infer the batch effect in single cell data. For the CCA, it's like PCA, it infers the CC1 and CC2. However, it doesn't really correct for any of the original observations in your original space. Okay, so it only aligns the CC1 and CC2. So for the MNN, it does output a corrected version of the gene expression matrix. However, that is bounded between 0 and 1. So it's not in the original range as well. So based on this, I think there will be still a lot of room for development, but I just want to point this out, and more details will be to follow. Okay, thanks.
Oh, yes, so to add to what Yuchao said, so most of these methods, so for example, in Harmony, they say very clearly that it's still primarily for visualization. So it's just for help you, so as I said earlier, it's just to help you do the visualization here. It's very hard to give you a batch corrected data in the original scale. So like Yuchao, I think Yuchao will talk more about that because of this confounding issue. Yeah.
Okay, it's my turn. Okay, thanks. So actually, I have 3 questions. So the first question is about this data denoising that you talk about. So as far as I understood, the difference between data imputation and data denoising is that data imputation is mainly concerning the 0 values. So you want to add some, offset to the 0 values. But denoising is more like opposite. You want to remove some positive values that are supposed to be noise. So as far as I understood from the figure that you show in one of the slides, before the denoising, you see a data set with excess number of 0 values. Then after denoising, those 0 values will be replaced with some small positive values. So in that sense, it's rather like, like adding some noise rather than denoising to me, because you're adding some values into the. So could you please explain a bit more about the rationale? Yeah.
So that's so first, some methods they call imputation, some methods they call denoising, but they are doing the same thing. So first, I'm just talking, when I say I will rather use denoising instead of imputation, is I just want to have a more precise description of what are people doing. Even then when they call imputation, they are also changing the non-zero values. I'm just saying in statistics, when you say imputation or intuitively, you are thinking that you are changing the zeros to non-zeros because you are thinking the zeros are missing. But here, it's not the same problem, so we like to call denoising better because you are removing the technical noise. But all these methods, though some are called denoising, some are called imputation, they are doing the same thing. And the other thing that we are changing zeros to some small non-zero values. You may think it's adding something, but here the thing is that the technical noise here is not additive. So you have Gaussian data. You may think, well, I have 5, and add some noise, it becomes 5.3, and that's kind of the noise. But here the noise is not from 5 to 5.3. It's from 5 to 0. So it's the noise of downsampling. So it's a different type of noise. So we are recovering. We are recovering from 0 to some. I would say we cannot recover from 0 to 5 because 5 is an integer. People first, typically when people do denoising, they are denoised to the normalized counts. So if you have if your true gene expression for one gene in the cell is 1% out of the total number of RNAs in the cell, you may observe a 0, and we want to recover from 0 to the 1%. So it's a different type of noise. It's not additive noise. So it's from 0 to a small number, but it's still doing the noise. It's still from 5.3 to 5. It's just a different type of noise.
Thanks. And the second question is related to the first one. So you talk about this special zero inflation feature in this single-cell RNA-seq data. So could you please illustrate more about why normally we see this kind of zero inflation in the single-cell data, or what is general pattern for all kinds of omics data, or any special reason for that? Why single-cell data?
So the zero inflation is not unique to single-cell RNA sequencing. If you look at if you look at proteomics data or the microbiome data, there are also a lot of zeros. And they also experience the zero inflation. So the zero inflation is a feature for counts data, the currently high throughput counts data. So the reason that you have zero inflation is because of downsampling. say you have 5 copies in the cell, and it's actually very hard to measure them. So what people do is that, let me turn to the page of this. So here, say you have like 2 copies. And then people do it if it's a first reversely transcribed into cDNA, then they amplify it so that they can sequence it better. So you have a lot of zero inflation is that first, for example, here you have 2 copies. It's originally 2. But when you do the reverse transcription, it has a efficiency rate of 30%. So that means that this 2 will like be very likely to be lost because you have a limited efficiency. And you see a lot of zero inflation because the single cell, the original RNA amount in one single cell is very limited. So it's very likely to be lost when this whole is not very efficient. So I think this kind of loss from RNA to cDNA is very hard to remove. And I think you can lose it also in the sequencing step. Because here you may have like here 2 copies, you now have 1, and then this 1 copy may be like amplified to 10 copies. But your sequencing rate may be like 10% if you don't have a very deep sequence, if a very deep sequencing depth. Then it can still be totally lost. So you will see that if some people have very high quality data, if the sequencing depth is like say is high, say like for example if the total if it's UMI data, if the total number of UMI is like 5,000, then like it will have the data will have less zeros because you have less loss during the sequencing step, but you still have some loss here. So the reason you see a lot of zeros is that this RNA get lost during the measuring step, and that's the technical noise. That's not like the Gaussian noise that you add something. So that's a common thing when you measure counts. Like for the microbiome data, you want to measure the number of microbiomes, and that's still kind of some counts, and it can be lost during the measurement step.
Okay, last question. So it's about this deep learning. So for deep learning, you need a lot of training data. So you need to have good data with labels of different cell types. So how easily you can get this good quality training data? I mean, in a single cell, normally you don't necessarily identify the cell subtypes beforehand.
Yes.
So what is the technique for?
So deep learning is a general technique where you use the neural networks to as your model, and it can do different things. When as the type of data you said is for classification, like I have all these images, and my training data have different labels for the images. Then for new data, I want to classify the new images based on the original labels. So that's one type of things that they can do. And this autoencoder is not for classification. It's for denoising. So it's a totally unsupervised method. So it's basically doing a dimension reduction. So you don't need to have the cell type labels because it's not doing classification. It's doing unsupervised learning, so you don't need that.
So yeah, right slide for me here. Autoencoder has not been used for classification, and you hit the point there. So the final error function, has it been that like should it classify properly the cell types based on autoencoder, or just denoising is the final goal here? But no one has. I don't see the classification getting better.
Oh, so you see, I hope I understand your questions correctly. Here we see classification better, but this denoising is not just for clustering. So here our goal, this loss function, is just for getting better quality data.
Right, but can it have some sort of, if you have cell type label and have error functions based on that?
So I think there are, so these are for denoising, but there are, I don't remember, I think it's, so there are some other work that they can do some cells have the labels, and they use that to guide you. So this is totally unsupervised. But you have some cells have some cell types that become some supervised. So this is totally unsupervised. You have some supervised thing, and you can make it semi-supervised. So kind of, I hope. So you can say like here I use this bottleneck layer to do classification for the cell types. So I don't remember the name of the work, but they are such type of thing.
And also the bottleneck layer, could it be used to identify top features, which is a problem in classification also? That's not a representation of the feature, it's some sort of latent layer, right? The bottleneck layer. It cannot be used to identify top features or important features which classify your sample. If your goal is just these bottleneck layers can help you do clustering, but it does not itself does not perform that well if you just use it to do clustering. There are work doing that. So I think you also need to have very good filtering step and also some other pre-processing steps to make it really work.
Okay, you said that you can separate like technical noise and biological noise in your X application. And could you be a bit more detailed how you want to do it? Because you said you're feeding public data, probably from different technologies and different biological entities into your network. And then how do you exactly separate technical noise from biological noise? Okay,
so I think that's two parts. So first, how are we possible to separate the tech? So how do we separate biological randomness from the technical noise? That's a long story. I think briefly, you can look at our paper if you're interested. But briefly, the idea is that we have a model for the technical noise. So for example, so we see the gap from here, but kind of we know that the technical noise can only be this much. So then the other part is the biological randomness that cannot be explained by the manifold. But how do we know that the technical noise is this much? Is that we have called a Poisson alpha model for the technical noise, and we verify that this model works well for most UMI data by looking at the spikings and some other things. And so that's the first part. So how do we separate biological randomness from the technical noise? Because we have a model for the technical noise kind of telling us how large the technical noise is based on your observation. And the second thing is that when we do transfer learning, the other data have different technical noise part. So that's why we first pre-trained the autoencoder using public data sets, which contains some feature that may be just the technical features of the other data sets. But then we trained the autoencoder again using our target data, and that can then these technical features that cannot be transferred to the new data, it will not be helpful for initialization. But when we train the autoencoder again on the target data, we can remove that part. That's the very high level.
You also have a model for the biological noise. So when you have like a Poisson distribution for the technical noise, what is your biological noise distribution? And how can you separate it? It's still not 100% clear.
So that's the thing. So this is the gap we see from the observed data to the manifold. And then we know the technical noise is this much, and that can help us identify the biological randomness, the gap from the true gene expression to the manifold due to biological randomness is this part. And this will be our estimate. So I think it's, I need a few more slides to explain that. You can look at the paper if that's.
When you use an autoencoder for denoising, couldn't you then, at the same step, learn the batch effect, for example, and skip the first part?
I think the SCBI so this paper, they are kind of incorporating, so they have a part to incorporate the batch effects into that. But one big problem, as I said earlier, the big challenge is that there is confounding between the batches and the unknown cell types, and that cannot be explained by, that cannot be easily solved just using autoencoder, because remember that these other methods, they try to filter out the cells that they think are kind of contain some specific features. So you need to have, so this one here, that kind of part is very hard to be incorporated in the loss function. When SVI do it, they are assuming that all the things, or the difference that you see is due to, between two different data sets are due to batches. And they just remove that to make these two data sets to be similar. But if there are unique features that represent just in one data set, it will be considered as batch effects. So I think that's the difficulty. But I know people are also working on that, yeah.
So I guess my question is, what kind of analysis do you think is fair after denoising? Because to me, it seems a little bit like it's amplifying latent structure and undersampled data, and I was just wondering, like.
I think so. The output of the denoise data is the same. It's the matrix of the same dimension as the original data. So I think in these papers, you can use it for doing doing clustering, and also construct the pseudotypes, and also visualize the marker gens. One thing that hasn't been solved well is that if you perform, say, differential testing on denoise data, it will create false positives, because originally your cells are independent to each other. After denoising, they get correlated. So we are writing a review paper to solve that. If you, we will put it soon on archive, yeah. But basically, doing inference on the denoise output, for example, differential testing, or like other, like Ron mentioned, like differential, like distribution analysis, mode analysis, that will give you some false positives. That's a hard thing, yeah.
And my other question is, how do you?
I think we can have the break right now, and if you have more questions, you can come to us during the break. Thank you.